Okay, so we are working our way through cellular respiration. The first thing we did was we talked about glycolysis, which is a catabolic pathway where we take one 6-carbon glucose and we split it apart through 10 reactions to produce 2 3-carbon pyruvates. In that process, we spend 2 ATP and we make 4, so it's a net of 2 ATP. We also reduce 2 electron carriers, 2 NADH molecules, which are later going to be sent to something called the electron transport chain. So this is an overall energetically favorable set of reactions, pathway. So now we have pyruvate. So if I was just to draw an overall of the electron transport chain, it would look like this. So here's glucose, and then here's the 10 steps of glycolysis. I'm not going to draw 10 arrows. And we make 2 pyruvates. And then we need to convert pyruvate to acetyl-CoA. Acetyl-CoA is the molecule that then starts the next big pathway, which is called the TCA cycle. The TCA cycle stands for the citric acid cycle. The next intermediate here is called citrate or citric acid. It's also sometimes called the Krebs cycle, or you'll see it called the TCA cycle. So this is, these are 2 of the pathways in cellular respiration. So we already said here we made some NADH, and then here we're going to make more NADH, and here we'll make even more NADH. We also are going to make a molecule called FADH2. So we'll make 2, 2, 6, 2 throughout the whole process. And all of these NADH and FADH2, these are all going to go to the electron transport chain. And the electron transport chain is where we are going to make most of our ATP. So we right now are here. We are stuck between, we've made the pyruvate, but the pyruvate does not enter the TCA cycle. So there's an enzyme that catalyzes, it's actually a group of 3 enzymes together that catalyze the conversion of pyruvate to a molecule called acetyl-CoA. Acetyl-CoA is what starts the citric acid cycle, or it's the molecule that enters the citric acid cycle. Pyruvate doesn't always have to go to the citric acid cycle. There are other metabolic fates for pyruvate. Pyruvate is what's called a metabolic branch point. So the reason that we would go through cellular respiration is when we have a lot of glucose available and we need to make energy. So there's other situations where we don't have a lot of glucose available, we don't need to make energy, and there are other things that we would do with pyruvate. Pyruvate is called a metabolic branch point because it has lots of different options. You can convert it into carbon dioxide through oxidative phosphorylation, which is what we're going to do down here in the electron transport chain. You can convert it to lactate. This is called homolactic fermentation. So pyruvate you can either convert to carbon dioxide and water. This is called oxidative phosphorylation. This happens in the electron transport chain. It is aerobic, which means it requires oxygen. Another option for pyruvate is to make something called lactate or lactic acid. This is called homolactic fermentation. This is anaerobic, so it does not require oxygen. Another thing we can do with pyruvate is alcoholic fermentation, which produces ethanol. Yeast and bacteria can do this. Our cells cannot. This is called alcoholic fermentation. Again, anaerobic. No O2. So we will mostly be focusing on this as we make pyruvate into acetyl-CoA to go through the citric acid cycle, to go to the electron transport chain, to undergo oxidative phosphorylation and make a lot of ATP. But that's not the only thing that happens to pyruvate. Our cells can undergo what's called homolactic fermentation, and other organisms can undergo alcoholic fermentation. So this is all on slides 58 and on, and it shows the conversion between this and then the situations where these different processes would happen. So lactic acid fermentation, for example, happens when there's no or low oxygen around. It's anaerobic. So you hear a lot about lactic acid buildup in your muscles when you're exercising. And what's happening is if you're exercising and you're using glucose to try to make energy, you're breaking down this sugar, you've made the pyruvate, but now you're out of oxygen. If you have plenty of oxygen, then you would just keep going through this path to make more ATP. It's an aerobic process. If there's no or low oxygen around, like a time of strenuous activity, then your muscle cells, your skeletal muscle, would convert this pyruvate into lactate. And that process is shown on slide number 59. And it shows the glucose being converted into pyruvate and then the pyruvate being converted into lactate. And what happens during this lactic acid creation is it also restores the oxidized electron carrier. So now we have more electron carriers to go through glycolysis. Glycolysis is an anaerobic process. You do not need oxygen to go through glycolysis. So lactic acid fermentation happens during times of strenuous exercise. It does produce hydrogen ions in lactic acid, and that's what causes your muscles to burn when you're exercising. It's not what causes your muscles to be sore the next day. Sometimes you hear people talk about that where they say their muscles are sore the next day and they say they have a buildup of lactic acid in their muscles. The lactic acid that builds up in your muscles is cleared from your muscles in about 30 minutes. So what makes your muscles sore the next day after exercising is something called DOMS, delayed onset muscle soreness. It's when your muscle tissue is actually breaking apart and then it's being regenerated. But lactic acid is what makes your muscles burn while you're exercising. It's not what makes your muscles sore later. And this happens in times of low oxygen, like strenuous exercise. If you have oxygen, then you would go through this process, which is what we are going to continue talking about when we continue down this road of cellular respiration and ultimately head to the electron transport chain for oxidative phosphorylation, which oxidative says oxygen right in the name. That does require oxygen. So we'll come back to pyruvate a few times in the next few weeks where we're talking about different metabolic branch points, where we're going to study a lot of these pathways. And then we are going to kind of see how they all are interrelated and how suppressing one pathway will upregulate another pathway and where there's some overlap. And pyruvate is going to come into play in a lot of those places. So glycolysis, glucose to pyruvate, but pyruvate cannot enter the next step. That is going to require pyruvate being converted to acetyl-CoA. So that process happens through a group of enzymes, 3 enzymes together that are going to convert pyruvate to acetyl-CoA. And that whole complex is called pyruvate dehydrogenase. It's actually 3 enzymes: enzyme 1, enzyme 2, enzyme 3. It's a complex of 3 enzymes. This is on slide number 93. I'm not going to try to draw it because it's all drawn out and shown nicely in your PowerPoint slides. But it shows what is happening and the role of each of these enzymes in the transformation of pyruvate to acetyl-CoA. This is also table 14.1 in your textbook. And it shows the role of the cofactor in each of these. So there's 3 enzymes and 5 cofactors. So to go from pyruvate to acetyl-CoA, there's 3 enzymes, a complex, and then there's 5 cofactors. And on table 14.1, or in your slides on slide number 94, it tells you the role of each of these cofactors in the transformation of pyruvate to acetyl-CoA. So this is called the pyruvate dehydrogenase multi-complex enzyme. And it is going to convert pyruvate and add this coenzyme A to produce acetyl-CoA. And then acetyl-CoA is the molecule that is going to enter the citric acid cycle. So this step right here is facilitating this reaction. So take a look at the slides. It's slide number 93. I used a few different textbooks just to show different images of these 3 enzymes and of these 5 cofactors and how this is catalyzing the reaction of pyruvate to acetyl-CoA.